I want everyone to imagine a domino cascade, where one domino knocks over two dominoes, and then those two dominoes knock over four, and then eight, and then 16, and so on and so on. You can imagine the vast number of dominoes that will fall over at the very end. Hundreds, thousands even, depending on how big you imagine this cascade to be. And to think, it all started with activating that one domino. In terms of relating this to the human body, we all have that one domino that causes an explosion of events just by activating it. These are called receptors, and there are many different types with different functions. One important type that we have is called the apelin receptor, which when activated by the apelin ligand, causes a cascade of events that is responsible for regulating our cardiovascular system and kidney function. However, in some cases, the apelinergic system has been linked with chronic heart failure, hypertension, and diabetes, very common and very serious diseases. Drugs can be used to negate these effects, but to create them, we need to study what its target is, and that target is how the apelin receptor and apelin ligand interact with each other. This is what my project consists of. We began by creating computational models of the apelin ligand and apelin receptor, and then formed a complex where the ligand was docked within the binding site of the receptor. With that, we moved on to creating a virtual environment where the complex was then embedded into a lipid membrane. A molecular dynamic simulation was then done to see how the receptor and the ligand interacted with each other. After running a 100 nanosecond simulation, we did RMSD analysis. RMSD tells us how a group of atoms deviate on average from the initial equilibrated structure. Our findings show that the apelin ligand stays fairly stable while interacting with the receptor and works towards an equilibrium. On the other hand, the receptor RMSD indicates that there is some sort of conformational change as this interaction proceeds. This leads us to exciting future prospects of the project. Through further analysis, we can deduce what specific atoms are changing in the receptor and to what extent. As well, we can look forward to analyze which specific parts of the ligand interacts with the receptor. Additionally, the simulation is currently being extended to 500 nanoseconds to tell us what further changes occur after the 100 nanosecond mark. And so, by moving forward with these aspects of the project, we gain the knowledge and mechanisms behind this interaction, which in the grand scheme of things aids in discovering and developing drugs to prevent the pathological conditions the system is linked with in the first place.